Okay, so I did my project on drivers of algal abundance in ditches on a South Central Florida ranch. And you have all probably seen pictures like this before. This is pictures of eutrophication happening in South Florida's waterways. The pictures on the left, gosh, now I'm all turned around. Yeah, the pictures on the left are pictures that I took in my hometown of Stuart, Florida, and you can see how crazy green the water is there. That's all algae. The picture in the middle is the same town, but a different part of the river. In 2016, we had a massive algae bloom that led to fish kills and other ecosystem destructions. And you can see there's a canal in the middle of that picture in the center, and it's completely green. It doesn't even look like water, but it is. Crazy. So these are toxic cyanobacteria algae blooms that lead to fish kills and other types of ecosystem horrors, I guess. And then on the pictures on the right, we have red tide, which is a different type of algae bloom. These are algae blooms that happen more so on the west coast of Florida rather than the east coast. And they are also toxic and can lead to fish kills and other types of die-offs. So, okay, gosh, they really ruined this slide. Okay, anyways, so eutrophication is caused by multiple things. It can be natural, which happens when sediment is deposited into a larger body of water, such as a lake or a pond. The streams deposit sediment, and with the sediment come nutrients such as nitrogen and phosphorus and other things that can lead to more phytoplankton and other types of growth. But humans have sped up this process a lot. They've added more nutrients to the soil in the forms of fertilizers. And whenever we go to the bathroom, we're also releasing nutrients into the ecosystem and into our septic tanks and our sewer systems and things like that. Humans have done another thing, and they've channelized a lot of streams. You can see in that center picture, that is Harney Pond Canal. That's one of the canals that we'll be talking about today. But it's completely channelized. It doesn't ebb and flow like a stream naturally would. There's no way for water to deposit sediment along the banks along the way. The water just flows very quickly and more directly into the mouth of the stream, which happens to be, for this stream, Lake Okeechobee. So yeah, so that's kind of what we're going to talk about today. The picture on the left shows a picture of Lake Okeechobee, which used to flow south naturally through the Florida Everglades. And the Everglades would act kind of like a filter for the nutrients passing through. They would soak up, the plants in the Everglades would soak up the nitrogen and phosphorus before it hit the coast. But the lake was dammed up about 100 years ago, and there were channels dug to the east and to the west to release lake water so that the dams didn't break. And so water was released west to Fort Myers, where we see those red tide algae blooms that we talked about earlier, and water is released east through the St. Lucie River to Stuart, which is where I'm from and why I love this. So anyways, that's where we see the toxic cyanobacteria algae blooms. So I wanted to look at what was happening a little bit upstream of Lake Okeechobee. So that middle picture, the red dot in the Lake Okeechobee watershed, that whole green area is part of the Lake Okeechobee watershed, but I was working in the red dot, which is Archbold's Buck Island Ranch. And the ranch is blown up in the picture on the right. And you can see there's a canal running through it. I didn't do a good job of it here. Maybe you can see my cursor. But the canal runs around the ranch for the top half and then through the bottom half of the ranch. And it eventually will lead into Lake Okeechobee. So that's what we're going to be talking about. So the ranch is cool because beef cattle ranching is the largest land use area in the Lake Okeechobee watershed. But as you can see, there's a lot of other land uses that are in the watershed. So we have a lot of citrus, that's in orange. There's urban areas in yellow, and there's dairy, sod, and all of those types of things. Also a lot of wetlands, and of course water. But Cattle ranches are the highest land use area in the Lake Okeechobee watershed, so a lot of people predict that they will still be the main focus for reducing nutrient loads within the watershed. So I wanted to measure algae abundance in ditches on a South Central Florida ranch, and I was hoping that that could help us better understand what was happening within our waterways downstream. So I figured out, with the help of Dr. Kelly, that we could measure chlorophyll A concentration as an indicator of algal biomass. We also, in my literature review, I found that there's a widely accepted positive relationship between high phosphorus and high algal growth within ditches within South Florida, not ditches. But we hadn't found any research on the relationships among chlorophyll A. I don't know how to go back. Nutrient concentrations like nitrogen and phosphorus, and biophysical characteristics like turbidity, temperature, conductivity, dissolved oxygen, and pH, all within ditches located on a working cattle ranch in South Central Florida. So there's been papers published on these things, but none of them have been happening. None of these relationships have been studied on ditches within a South Central Florida ranch in the Lake Okeechobee watershed. So that's where we come into play. So I figured out we could do this. We had 19 ditch sites on Buck Island Ranch that we were looking at. We had 8 of them located along the Harney Pond Canal that we were talking about earlier, and those ditch sites were a little bit wider and deeper. They had water pretty much year round, and a lot of that water was mixed with the Harney Pond Canal. So these are the internal sites, or the internal side of the ditch. So we get runoff from the ranch as well as a mixture of Harney Pond canal water in those sites. In the dry season, a lot of this is pumped, so most of the water is from the Harney Pond Canal, and then in the wet season, it's not as pumped and most of it is runoff from the ranch. So then the middle column shows hickory branch sites, which we had 3 sites that we categorized as hickory branch sites, and those were located in a stream that runs through the ranch. And so the stream runs off the Lake Wales Ridge, which is located in Central Florida, and it flows through citrus groves, some other ranches, some wetlands, and other land use areas before it reaches Buck Island Ranch, where it continues through the ranch and ends in Harney Pond Canal. So this stream obviously doesn't really look like the ditch sites, and it is more naturally flowing. It kind of ebbs and flows more so like a stream. And then our last category was internal Buck Island Ranch ditches, and these ditches are a little bit shallower. Sometimes they dry up in the dry season, and in the dry season, they're mostly pumped from Harney Pond Canal. So water is pumped all the way through our ranch so that we can maintain water for the cows and for the pastures. So I looked at 3 main research questions. Gosh. This used to be a PowerPoint and I changed it and now it's messy. Anyways, it's a Google slide now. So, okay, our first research question was, how do nutrient concentrations like nitrate, ammonium, total nitrogen, total phosphorus, and orthophosphate relate to chlorophyll A concentrations? And then how do biophysical water quality parameters relate to chlorophyll A concentrations? Our second research question was, will chlorophyll A and water quality parameters differ in ditches categorized as Harney Pond sites, hickory branch sites, or internal Buck Island Ranch sites? And then our third question was, which water quality parameters would explain the most variation within chlorophyll A concentrations? So I thought that higher nutrient concentrations and higher light availability and higher temperatures would all lead to higher chlorophyll A concentrations. I also thought that conductivity and pH would be insignificantly related to chlorophyll A, and that more turbid waters would increase nutrient concentrations, and therefore chlorophyll A concentrations. So then I thought that higher chlorophyll A in internal Buck Island Ranch ditches would happen because of legacy nutrient application and low flow rates within those sites. So Buck Island Ranch used to be some of our internal pastures were fertilized with phosphorus until the 80s, but the phosphorus doesn't go away overnight. And so we still are seeing legacy phosphorus in our soil and in our water within those ditches. And then current nitrogen application happens every few years whenever it's needed for the pastures. So yeah, So then I thought that total phosphorus, total nitrogen, temperature, and turbidity would explain the most variation within chlorophyll A concentrations. So, like I said earlier, we had 8 Harney Pond sites, and these sites are located along the Harney Pond Canal, which runs through Buck Island Ranch. You can see those are the little plus sign things going on there. And then we have the Hickory Branch sites, which are located in the lower left-hand corner of the map. They're SW1, SW2, and SW3, and those are all along the stream as it leads to Harney Pond Canal. And then we have the internal Buck Island Ranch sites, which are the rest of the sites that are more internal in our pastures where we have cows and things like that. So we decided to sample monthly for 4 months. We collected biophysical data using a YSI, and then we collected nutrient data using colorization protocols on a spectrophotometer. And then these were all things that we had done at the lab in the past, but we didn't have a working chlorophyll A protocol. So I worked with Dr. Kelly to establish one of those at our lab. So to do this, we figured out we could grab surface samples in the field and then bring them back to the lab where we would filter with a vacuum pump and then wrap those filters in aluminum foil and store them in a freezer until we were ready to analyze them. So to analyze the filters, we would extract them using a 90% acetone solution and soak them overnight in the fridge and then read them on a spectrophotometer the next day. So wow, wait, it didn't have my data analysis slide. Oh, there we go. Okay. So for data analysis, we log transformed chlorophyll A, and so we added 0.001 to all the values so that we could log the zero values, the true zero values. And then we used a Pearson correlation test to see which variables were highly correlated to one another, and then which variables were correlated to chlorophyll A. We used a Kruskal-Wallis test for one-way ANOVAs and an AIC model selection of 30 models with the covariates total phosphorus, total nitrogen, temperature, turbidity, site category, vegetation cover, and water flow. So we removed the highly correlated covariates and we included a null model in this. All right, here we go. So we found that chlorophyll A concentrations overall were positively related to phosphorus, and that the relationship was significant. And that's what we were expecting. But what we weren't expecting was total nitrogen and nitrate to be negatively related to chlorophyll A. This is something that we did not foresee. And yeah, I have a lot of ideas on why this is happening, but we don't have all the time in the world here. So cool stuff. Anyways, then we found that chlorophyll A was positively related to turbidity and negatively related to dissolved oxygen, and both of these relationships are significant and somewhat expected. All right, so then we found that there was high total phosphorus and high chlorophyll A in internal Buck Island Ranch sites compared to Harney Pond and Hickory Branch sites. And we also found that when we split up the nutrients by sites, that nitrates and total nitrogen were extremely high in Hickory Branch compared to Harney Pond and internal Buck Island Ranch ditches. We also split the biophysical data up by site and found that temperature, conductivity, and pH were highest in Harney Pond sites and lowest in internal Buck Island Ranch sites. And then we saw that dissolved oxygen was highest in Hickory Branch, probably because it's a more flowing stream. And then we found that turbidity was highest in internal Buck Island Ranch ditches. So then we looked at the model selection for the most explanatory variables for chlorophyll A concentrations, and we found that total nitrogen and site category was the model that explained the most variation within chlorophyll A. So it has a weight of 0.38, which means that it explains 38% of the variation within chlorophyll A. So total phosphorus and site category was also a significant result, or a somewhat important result, as it had a delta AIC of less than 2. And it was weighted at 0.19, meaning that it explains about 19% of the variation within chlorophyll A. And then site category by itself was a significant result as well, and explained about 17% of the variation within chlorophyll A. So I included the null model on here just so that we could see that Its AIC, its delta AIC is way higher than 2. And if our stuff was just due to chance, if our data was due to chance, the null model would have a delta AIC of less than 2, hopefully. Okay, so then I split everything up by site because that's what the analysis said was the most explanatory variable. So I split everything up by site and looked at total phosphorus and total nitrogen by site. So here, nothing is as significant as it was before, but you can see that the axes, the x-axes specifically, are very different. So if you look at in the Harney Pond column, you see on the bottom there's total nitrogen and log of chlorophyll A, and this axis ranges from about 0 to 5, whereas the one right next to it in Hickory Branch, the nitrates in Hickory Branch, this axis ranges from about 8 to 13. So total nitrogen in Hickory Branch is way higher and showing a completely different version of the graph that you would see in the Harney Pond sites. And same with the internal Buck Island Ranch ditches, that axis is showing a whole different grouping ranging from about 0 to 11. So this graph is significant. The one that's showing nitrate, total nitrogen, and log of chlorophyll A within the internal Buck Island Ranch ditches, this is the only significant graph based on p-value in this grouping. And that was interesting because we found that overall nitrates had a negative relationship with chlorophyll, but when we look specifically at internal Buck Island Ranch ditches, nitrates have a positive relationship with chlorophyll. Pretty cool stuff. So I found that based on the South Florida Water Management District, concentrations of chlorophyll that are higher than 40 parts per billion are considered to represent a moderate algae bloom. So I looked at the algae blooms within our ranch and found that we had 3 sites that had algae blooms according to district, district protocols, whatever. And anyways, so we had one site that had about 300 parts per billion for chlorophyll, and that is a crazy high concentration. The water was super green in that site, and it was pretty cool. Anyways, it's important to point out that all of these are internal sites, and I think these are less flowing. They have cows in them. They're like all pumped water from Harney Pond Canal at this point. This is all winter stuff, so it's dry season. So yeah, what's going on there. And then I also wanted to point out that all 3 of the Hickory Branch sites had the lowest chlorophyll levels. They ranged from 0 to 2 parts per billion. These sites also have cows in them, but the site is flowing a little bit faster and had super high nitrates and a lot lower phosphorus than the internal sites. All right, so yeah, we already saw that slide. All right, so why does this matter? Water is a super important part of every ecosystem, right? Everything depends on water, from agriculture to native animals, native plants. And so I think it's important for us to try to protect our natural systems. And in doing so, we can protect agriculture and we can protect economies, especially coastal economies that depend so heavily on tourism. If we have super harmful algae blooms all the time, maybe we won't get enough tourists to make up for our economy. And yeah, it would be a little bit taxing on that. So there are some limitations that we had during our experiment. And so we weren't able to measure dissolved organic carbon, and we couldn't account for pumping. I wanted to look at specifically runoff from the different pasture types within the ditches, but we couldn't really do that in the dry season. So some of the water is runoff in the internal ditches, but most of it is pumped from Harney Pond. And so I think that's important to note. And then we also did not account for parasitism, macrophytes, and grazing by zooplankton, which can all affect phytoplankton counts. So we also only looked at 4 months, and so we're not accounting for seasonality. We're not accounting for those super high temperatures that we see in August and September. We're not accounting for hurricanes and rainy, stormy events that happen during the wet season. And we're also looking at one ranch within the South Central Florida region. This can't be representative of every single ranch here, so um, just keep that in mind. So yeah, future directions, I would love to look at seasonality a little bit more and see what's actually going on in the summer when we see a lot of those harmful algae blooms on the coast. So what's going on upstream of those coastal systems in the summer. And then phytoplankton can live for like 3 to 28 days, and so maybe we should look at chlorophyll A concentrations from this month with nutrient data from last month to see how the phytoplankton grow over time as nutrients increase or decrease or change. And then the real world application of this research, I guess, would be payment for ecosystem services. That we already have a Northern Everglades payment for ecosystem services where ranchers are actually paid to hold back water, to board up their culverts on their ranches and hold back water to keep it from releasing into the Lake Okeechobee watershed. And so maybe we should look at seeing when chlorophyll is highest and when we have these algae blooms on ranches, and maybe we should be holding water back when we have algae blooms. So yeah, that is all. Thanks to all these crazy people. They're the best, and I wouldn't be here without them.